DNA damage and causes. The four types of factors that cause DNA damage are hydrolysis, deamination, alkylation, and oxidation. Cause 1. Hydrolysis. DNA consists of long strands of sugar molecules called deoxyribose that are linked together by phosphate groups. Each sugar molecule carries one of the four natural DNA bases adenine, guanine, cytosine, or thymine, A, G, C, or T. The chemical bond between the DNA base and its respective deoxyribose, although relatively stable, is nonetheless subject to chance cleavage by a water molecule in a process known as spontaneous hydrolysis. Loss of the purine bases, guanine and adenine, is referred to as depurination, whereas loss of the pyrimidine bases, cytosine and thymine, is called depyrimidination. In mammalian cells, it is estimated that depurination occurs at the rate of about 10 purine bases lost per cell generation. The rate of depyrimidination is considerably slower, resulting in the loss of about 500 pyrimidine bases per cell generation. The baseless sugars that result from these processes are commonly referred to as AP sites. Loss of purine base guanine and adenine is referred to as depurination. And again, I will be reading this. DNA consists of long strands of sugar molecules called deoxyribose that are linked together by phosphate groups. Each sugar molecule carries one of the four natural DNA bases adenine, guanine, cytosine, or thymine. A chemical bond between a DNA base and its respective deoxyribose, although relatively stable, is nonetheless subject to chance cleavage by a water molecule in a process known as spontaneous hydrolysis. Loss of purine base guanine and adenine is referred to as depurination, whereas loss of pyrimidine bases cytosine and thymine is called depyrimidination. In mammalian cells, it is estimated that depurination occurs at the rate of about 10 purine bases lost per cell generation. The rate of depyrimidination is considerably slower, resulting in the loss of about 500 pyrimidine bases per cell generation. The baseless sugar that results from these processes are commonly referred to as AP sites, apurinic, pyrimidinic. They are potentially lethal to the cell as they act to block the process of DNA replication but are repaired, efficiently repaired in a series of enzyme catalyzed reaction collectively referred to as the base excision repair or BER pathway. In fact, AP sites are intentionally created during the course of BER, then cause to deamination. In bases that make up DNA are also vulnerable to modification in their chemical structure. One form of modification called spontaneous deamination is the loss of an amino group NH2. For example, cytosine, which is paired with guanine, is normal. Double-stranded DNA has an amino group attached to the fourth carbon of the base. When that amino group is lost, either through spontaneous chemical or enzymatic hydrolysis, a uracil base is formed and a normal GCA-DNA base pair is linked to a pre-mutagenic UG base pair. Uracil is not a normal part of DNA. The UG base pair is called a pre-mutagenic because it is not required before DNA replication. A mutation will result. During DNA replication, the DNA strands separate and each strand is copied by a DNA polymerase protein complex. On one strand, the uracil will pair with a new adenine, and while on the other strand, the guanine will pair with a new cytosine. Thus, one strand DNA double strand contains a normal GC base pair, but the other double strand has a mutant UA base pair. This process is called mutation fixation, and the mutation of the G to A is said to be fixed, meaning fixed in place, not repaired. In other words, the cell now accepts the new mutant base pair as normal. It is estimated that approximately 400 cytosine deamination events per genome occur every day. Clearly, it is now very important for the cell to repair DNA damage before DNA replication commences in order to avoid mutation fixation. One cause of normal human aging is the gradual accumulation over time of mutation in our cellular DNA. Then alkylation, one type of base alkylation occurs when a reactive mutagen transfers an alkyl group, typically a small hydrocarbon side chain, such as a methyl or ethyl group denoted as CH3 and C2H5 respectively, to a DNA base. A nitrogen atom of the purine bases N3 of adenine and N7 of guanine and the oxygen atom of guanine are particularly susceptible to alkylation in the form of methylation. Methylation of DNA bases can occur through the action of exogenous environmental and endogenous intracellular agents. For example, exogenous chemicals such as dimethyl sulfate used in many industrial processes and formed during the combustion of sulfur-containing fossil and N-methyl N-nitrosamine, a component of tobacco smoke, are powerful alkylating agents. These chemicals are known to greatly elevate mutation rates in culture cells and cause cancer in rodents. Inside every cell is a small molecule called the S-adenosyl methionine or SAM. S-adenosyl methionine or SAM, which is required for normal cellular metabolism, is an endogenous methyl donor. The function of SAM is to provide an activated methyl group for virtually every normal biological methylation reaction. SAM helps to make important molecules such as adrenaline, a hormone secreted in times of stress, creatine, which provides energy for muscle contraction, and phosphatidylcholine, an important component of cell membranes. However, some can also methylate inappropriate targets such as adenines and guanines. Each endogenous DNA alkylation damage must be continuously repaired, otherwise mutation fixation can occur. Then, oxidation. Oxidative damage to DNA bases occur when an oxygen atom binds to carbon atom in the DNA base. High energy radiation like X-rays and gamma radiation cause exogenous oxidative DNA base damage by interacting with water molecules to create highly reactive oxygen species, which then attack DNA base at susceptible carbon atoms. Oxidative base damage is also endogenously produced by reactive oxygen species released during normal respiration in mitochondria as cells' energy factories. Humans enjoy a long lifespan, thus it would seem that healthy DNA repair proficient cells could correct most of the naturally occurring endogenous DNA damage. Unfortunately, when levels of endogenous DNA damage are high, which might occur as a result of inactivating mutation in the DNA repair gene, or when we are exposed to harmful exogenous agents like radiation or dangerous chemicals, the cell's DNA repair system become overwhelmed. The lack of DNA repair results in high mutation rate, which in turn may lead to cell death, cancer, and other diseases. Also, if the level of DNA repair activity declines with age, then the mutational burden of a cell will increase as you grow older. Then DNA lesions and their causes. Missing base. Removal of purines by acid and heat under physiological conditions, 10 raised to 4 purines per day per cell in a mammalian genome. Removal of altered base uracil by DNA glycosylases, then altered base ionizing radiation alkylating agents like ethyl methane sulfonate, then incorrect base mutation affecting 3' to 5' exonuclease proofreading of incorrectly incorporated bases, bulge due to deletion or insertion of a nucleotide. In intercalating agents that cause addition or loss of nucleotide during recombination or replication, then linked pyrimidines. Cyclobutyl dimer, usually thymine dimers resulting from UV radiation, single or double strand breakage of phosphodiester bonds by ionizing radiation or chemical agents. Bleomycin, then cross-linked strands, covalent linkage of two strands by bifunctional alkylating agents, mitomycin C. Then 3' deoxyribose fragments, disruption of deoxyribose structure by free radicals leading to strand breaks.